Today we're going to be talking about Chapter 8, Section 4, which covers the topic of transcription. Before we get started, make sure you have the corresponding Power Notes sheet. It has the central dogma at the top, and then a place for you to outline differences between DNA and RNA, and then this really handy figure for you to label at the bottom with the different components of transcription. So what we're talking about in transcription is going to be how we go from a gene, which is a segment of DNA, and we use that gene to make a single-stranded RNA molecule, as you can see outlined here in this diagram. This is going to be happening inside the nucleus, so we're just focusing here on transcription today. Translation will be the focus of our next presentation, and that will happen outside the nucleus. So again, we're just focusing on this initial part of transcription happening inside the cell's nucleus. When we are talking about transcription, this is going to be a part of what is called the central dogma of biology. And the central dogma is outlined here in pictures. You can see that DNA is the molecule that stores our genetic information. The process of transcription, as you can see in the arrow, is going to take DNA and use it to make mRNA. mRNA are going to be the instructions for making a protein. And then the final stage of the central dogma is going to be translation, where you are taking that strand of mRNA, the instructions, and we're using it to actually build the protein. So the central dogma, simply stated, is going to describe the flow of information in a cell. The information is stored in DNA, it's used to make RNA, and finally turned into proteins. Now we need to talk about the differences between DNA and RNA because they're both a really important part of the process of transcription, and they each have their own unique structures and their unique functions. So first of all, RNA is going to be a single-stranded molecule, like you see here on the left. It's not that double helix like we would expect in DNA, it's just a single strand. Now sometimes it does fold back on itself, and there will be some sections of an RNA molecule that are paired together, but it is still just a single strand. DNA, on the other hand, is a double helix. RNA has a different sugar from DNA. RNA has ribose. You can see that pictured on the left. DNA, we've already talked about this, has the sugar deoxyribose. You can see that the only difference between these sugars is going to be what is attached to this carbon. It's an OH group or a hydroxyl group in ribose, but if you look at deoxyribose, it's missing the oxygen, hence the name deoxyribose. And finally, there is a difference in the nitrogen bases. RNA has the base uracil, DNA has the base thymine. Those are the only differences. The rest of the bases, the cytosine, adenine, and guanine, are going to be the same in both RNA and DNA. But that uracil, you can see here on the left, differs very slightly from a thymine, which you see on the right. It's a CH3, which is a methyl group in the thymine, and it's just an H bonded to this carbon in the uracil. But that difference does impact how those bases are going to form together and how they're going to function within the molecules. Now we have another enzyme to learn about here in transcription. Transcription is going to be catalyzed by an enzyme called Rna Polymerase, just like we learned in DNA replication. A polymerase is something that's building a polymer. Well, if we have RNA polymerase, the polymer being built is going to be RNA. So during transcription, the information from one single gene, we're not talking about the entire DNA strand, start to finish, just one gene, one segment of that DNA. That information is copied into a strand of mRNA. So here you can see if we have a strand of DNA, there's a beginning, which we call a promoter. There is the end of a gene, which is a region called the terminator, and just this one segment of DNA would be the gene that is made into RNA. And that RNA is, again, carrying that message from DNA, which is inside the nucleus of the cell. It's going to take it outside of the nucleus into the cytoplasm to a ribosome where it can be used to make protein. So let's talk about each step in a little more detail. Our first step is going to be RNA polymerase and other proteins called transcription factors. They're going to form this complex, which is basically just a collection of proteins at that promoter region. Now remember, the promoter is going to be the beginning of a gene. So that is the region right here, and you can see along that promoter region, this would be a strand of DNA. We have all of these other colored circles and shapes. These are different proteins that all assemble together in order to form this transcription complex. Now, you do not need to know all of the names of the different transcription factors. There are way more transcription factors than we have time to really discuss. But they are specific to different types of cells, different types of genes, and they're important for controlling how genes are expressed. Our second step here is going to be RNA polymerase. Remember, we said that was the enzyme that's making the RNA. It is going to separate those DNA strands, and as the strands are separated, RNA polymerase will add complementary Rna nucleotides to the exposed bases. So this is how we make our new strand of RNA. Now you'll notice that that DNA double helix is going to form again right behind that RNA polymerase. So as the RNA polymerase is moving, it's moving from left to right in this picture, so in this direction. As that RNA polymerase moves along the strand of DNA, notice that it goes back to its normal double helix shape at the end. And this is going to continue until the RNA polymerase reaches the end of that gene, which again we said was the terminator. So we have a strand of DNA, and when I say gene, I just mean one segment of DNA that's the code for a particular protein. So in our chromosomes, we have long stretches of DNA, but that DNA in a chromosome could have hundreds or thousands of different genes. So when we are transcribing a gene, we are just transcribing the one section that has the instructions for the one protein that we need to make. So you can see here, this is another view of the whole process where we have the promoter, the RNA transcript is being made based on this sequence of DNA, and it will continue until it gets to that terminator sequence. And this would be the RNA polymerase. RNA polymerase's job is to take RNA nucleotides and match them up to the exposed DNA bases. So now you have this diagram on your Power Notes sheet. Go ahead and pause this video right now and see if you can label all of the boxes on your own. And then once you think you have all the answers, go ahead and hit play and check your work. So our first box here is pointing to the yellow thing, which is going to be our RNA polymerase. You could also call it the transcription complex, that whole collection of RNA polymerase and other proteins that are working together to cause transcription. Box number 2, that is our template strand of DNA. Notice that this red strand of DNA is the strand that's being used to make the RNA, and that's why we call it our template strand. Box number 3, that will be the promoter. Remember, that's the beginning of the gene. That is where the RNA polymerase is going to start transcribing the gene. Box number 4, that's the end of our gene, or the terminator. Box number 5, this is going to be our mRNA. Now you'll notice that it's called pre-mRNA here. That's because it actually needs to be processed a little bit before it can be used, but we're not really going to be getting into the details of that process today. And then finally, box number 6 is going to be the RNA nucleotides. That new strand of RNA, the green strand, has to be built out of something, and being RNA, its building block is going to be nucleotides of RNA. So ribose, sugar, adenine, guanine, cytosine, and uracil would be the nitrogen bases of those RNA nucleotides. So let's talk about the 3 different types of RNA, because there are different types of RNA that are used for different functions. The first one is what we've already talked about. That's the messenger RNA, or mRNA. It carries instructions for making proteins from the nucleus to the ribosome, and that's what we've already discussed. It's the message. It's taking the message from the DNA to the ribosome, which reads the message in order to make a protein. Ribosomal RNA, or rRNA. The ribosome itself, remember we talked about ribosomes not being membrane-bound organelles. Well, that is because the ribosome is just made out of RNA and some protein. So the ribosomal RNA is the RNA that literally makes up the structure of the ribosome. And then it's held together with various proteins, which also is an important component of a ribosome. And then finally, our third type of RNA is called transfer RNA, or tRNA. And the reason why it's called transfer RNA is because it's transferring or carrying an amino acid to the ribosome for use in making a protein. Now, obviously, we did not discuss that today. That's going to happen in the next part of protein synthesis, which is called translation. So the rRNA and tRNA are both important in the process of translation. The only type of RNA that we had in transcription was the messenger RNA, or mRNA. Now, transcription is similar to replication in a lot of ways, so make sure that you're not confusing the two. They both are going to involve complex enzymes and complementary base pairing. So there's always going to be some kind of enzyme that is opening up a strand of DNA, adding complementary bases. So what's the difference here? Well, the end result is really the main difference. In replication, remember, we're making a copy of the entire strand of DNA. Transcription is only going to be targeting one specific gene, not the entire strand of DNA. It just takes the message from one particular gene. Replication is also only going to make a single copy of the DNA. When your DNA is copied in your cells, it's only copied once, and each copy of DNA goes into a different daughter cell. And then eventually, if that daughter cell divides, then the DNA would be copied again. But every time replication happens, it's only making a single copy. Transcription, however, is making many copies. When a gene is being expressed, transcription is going to make lots of copies of mRNA, so there's lots of instructions available for making the protein. This image is a really good example of how one strand of DNA can be transcribed many times. This segment of DNA, you can see the DNA is this thread in the middle. This segment of DNA is a gene, and notice all of this RNA being made from that one gene. So if you are trying to express a gene and make a lot of protein, it can be transcribed many times all at once in order to make enough protein for the cell. So to end up today, this is going to be the section 8.4 quiz from your textbook. So go ahead and think about what we have talked about during the process of transcription. Take a look at these 5 questions and see if you can answer them. Make a note of your answers and we will discuss them in class. Hope you found this helpful.